and we'll also identify the component and the reaction setup of a typical PCR. So before we go, I want to give us a scenario now. So let's assume one of us now is a forensic scientist, and then there was a crime scene where maybe a theft or an accident. Then one of us, who is the forensic scientist, received a drop of blood from that crime scene. And then he went to the lab. He extracted the DNA just like we did yesterday. But that amount of DNA in that drop is too little to analyze directly. What does he or she, what does he do next? That's where we enter our PCR. So with the PCR, you can amplify that little amount of DNA that we have. We can amplify it and get billions of copies within hours. What is PCR? You can think of your PCR like a photocopy, yes, like a photocopier machine, like a photocopy machine. So PCR, polymerase chain reaction, is used to amplify a specific DNA sequence, like a specific region, and then produce millions to billions of copies in hours. And this is very essential for gene detection, expression studies, sequences, and diagnostics. So I'll quickly talk briefly about DNA replication, because DNA replication is in vivo. It happens inside of us, but while PCR is in vitro, we do this on the bench. But both PCR and DNA replication, they are doing the same thing. PCR is also amplifying, making several of the same thing DNA replication also does inside of us. So DNA replication is making a new, is when a cell copy the old strand plus the new. So you make when you have a new strand, you know our DNA is double stranded. So when replication happens, the old strand, then another new ones will be formed with the help of the enzymes DNA polymerase, our primers. This will now help to add a new one to the existing one. Then we now have another copy. As we go on, you are going to see how it is done. So, like I said, PCR is built upon the principle of in vivo DNA replication. And in DNA replication, we make use of enzymes that create DNA molecules by assembling the nucleotides. That will be the building blocks for the DNA. So we have the early cases. This is DNA replication. I just want to refresh our memory before we go to the bench. That's the PCR. We have the early cases. These are the ones that will separate the strands of our DNA double helix. Then we have the topoisomerase. These are the ones that will regulate the unwinding and the underwinding of the DNA. Then we have our SSB proteins. That's the single strand binding proteins. They will be the one to keep the DNA single stranded after it has been melted by the helicase. And then we have the telomerase, we have the primase. So these are all what happen for during. These are the enzymes that aid DNA replication inside of us. But for PCR now, we make use of DNTPs. This DNTPs contains our DTTP, DCTP, DGTP, and DATP. So the T is thymine, the C is cytosine, guanine, and adenine. They will make use of polymerase. Inside of us, we make use of DNA polymerase, but now it has been scientists was able to make it inside to make it in what we can now order with a tag polymerase. So this tag polymerase can now undergo the heat. Then we'll make use of our primers. Primers are short nucleic acid strands. They will be the one to add the nucleotides So they serve as the starting point for the DNA synthesis. And primers are made, they are made to complement the particular region or tag. Let's say now the DNA sample we all have with us now. My own is I want to amplify the region that codes for my uh we have our old gene with us, the old DNA, but we are just interested in a particular segment. That's a gene now. We have our old DNA, but we are particular we want to amplify a particular region. So for me now, I want to amplify the region that codes for my height. So you can say, okay, you want the gene that codes for your metabolism. So the primers I'm going to get now, they will complement, they will be designed particularly for that region. So that's what the primers does. Then we have our magnesium chloride and buffer, and we have our thermocycler, and then the template DNA. But with advancement in technology now, all these DNTPs, tag polymerase, MGL, and buffer, everything has now been added together. We now have a master mix. We can now buy a master mix that now contains our DNTPs, our polymerase, our magnesium chloride. Instead of getting them separately, we now have a combined mix that we call the master mix, that we now buy. Instead of saying, oh, I want to buy DNTPs, I want to buy polymerase, I want to buy magnesium chloride, I want to buy buffer, we now buy master mix. That master mix now comprises of all of these things. Now consists of all of these things. It's only your primers, your template DNA, your master mix, and your nucleus filter they are going to mix together. When we get to the reaction mix, we'll do that. Then you make use of a thermocycler. This is what we distribute the temperature per cycle to make the copies for the DNA. So the principles of polymerase chain reaction, like the name, chain reaction nature. That's where the power of the PCR lies. So each cycle takes the product of the previous cycle and then uses them as a new template for further application. We have 2 strands. After the 4th cycle, we will now have 4. 1 old and 1 new. Then it goes another cycle again. Then it goes another cycle. And if you are doing conventional PCR, your cycles will be at least 30 cycles. Now imagine that in 30 cycles. We already have millions of that particular region. So the newly synthesized strands now becomes a template for the next cycle. Then the amount of that target DNA literally doubles with each cycle, creating a geometric progression. Then it goes like that. It goes like that with the thermocycling condition of denaturation, annealing, and extension. So denaturation is when the strands separate. Annealing is where the primers now come. Oh, okay, this is the region you want me to bind to. The primer now locates them and then starts binding to the complementary strand, to the complementary bases, nucleotides. Oh, okay, this is where I'm going to. They start binding to it. When they are now done binding, extension, we now add the polymerase to extend the nucleotides. So by that, 4th cycle goes again the 2nd cycle, 3rd cycle, till the 38th cycle. If you are doing a real-time PCR, you use 40 cycles, sometimes 45 cycles. So that is the principles of PCR. So now the PCR cycle, step 1 is denaturation. I already said this. It separates the double-stranded DNA template into 2 single strands. It separates them by heating. And the temperature is between 94 to 98 degrees Celsius. But usually use 95. 95 for 30 seconds. So that thermal energy will now overcome the hydrogen bonds between the complementary bases. So it will now cause the DNA helix to unwind and separate. So that's the denaturation. Then annealing now. The primers bind. They anneal to their specific complementary sequence. Oh, the primers I made is for my for what's it called, to code for my height. Once I make the primer, I get my primer. So your primer come in a lyophilized form. Is always like in a jelly, like in a tube. Then you reconstitute it with nucleus-free water. So let's say when you get your primer, then you add, let's say the N mole is always written in the primer sheet. So let's say the N mole is 57.62. The amount of nucleus-free water you are going to add, you multiply the 57.62, the N mole, multiply by 10. So that will be 576.2 if I'm correct. That's the amount of nucleus-free water you are going to add inside the primer that you order. So once you get that, once you add, reconstitute it, that is still a stock solution. You still cannot use that primer like that. It's too concentrated. You have to make it into a working solution. So you make it into a 10 nanomolar. And for you to make that, you take that stock solution, you take 10 microliters of it inside another microcentrifuge tube, and then add 90 microliter of your water. So that is how you make your primer. I hope we are making that already, because we are going to need it when we want to set up our PCR with our DNA. So now that we have made our primer working solution, the stock solution, we'll keep it in the freezer -80 till whenever we need it again. But our working solution is what we'll be needing now. 10 microliter of our stock and 90 microliter of our working 10 microliter of our stock and 90 microliter of your nucleus-free water. And the temperature for your annealing, it depends on your primer. The annealing temperature is not generic. It's not something that is like your initial denaturation, like your extension. Annealing is if the primer I'm using that coats for my height, if after I calculate the temperature, I'm still going to talk about how to calculate your temperature for your primer. If after I calculate my temperature for my primer now, and then it is 55, I'm going to use 55. If Christiana calculates her own and it's 60, she's going to use 60. So annealing temperature depends, is specific to the primers used. And for you to calculate it, so for you to calculate your annealing temperature for your primer, so we have I think after this class, like after I'm done sharing the slide, then I can now share my screen as well, and then we'll do the website together. But there is a calculator we use, NEBTM calculator. So I'm going to take us through how to use the calculator to calculate the annealing temperature. So we use NEBTM calculator. And also there is also a mathematical, like a simple formula that we can use as well. That's TA. Yes, TA, that's your annealing temperature. Is always written in the primer sheet. What I will do is I'm going to send a picture of how a primer sheet looks like, so you know all the information that is, that is going to be, or that is always inside the primer sheet. Then annealing, then extension. Now the extension is, I already said it earlier. So if you look at the picture, that's when the DNA polymerase now adds the nucleotides. After the primers already bind to it, then it now started you start to add the nucleotide to it, and the temperature is usually 72 degrees Celsius for 60 seconds. But these are just the 3 steps: the denaturation, annealing, and extension. But when you want to set it on your thermocycler, you start with an initial denaturation. This is to get the machine to the temperature that you want. So we usually set 95 for like 10 minutes. So we believe in 10 minutes, the machine will get to that 95 degrees Celsius, that temperature, that heat, before the denaturation now starts, then the annealing, and then the extension. But the denaturation will only happen in one cycle. What will happen in the 30 cycles we are doing is just our denaturation, annealing, and extension. Then there's always a final extension too, 72 degrees Celsius for 5 minutes. So that's what happens during extension. So now we want to prepare we can bring out our DNA now. We'll get our Eppendorf tubes or PCR tubes, the 200 microliter one, those small tubes. We get them, and then we have just one sample. We'll take one of them, label it with your initials, and then you take your. So we are making a 20 microliter reaction now. Everything that is going to be inside that PCR tube will not be more than 20 microliters. So you can draw a table, write components, then the next row write 20 microliter reaction. So under the component, write your template DNA, then write your forward primers, then reverse primer. The primer always come in a pair, forward and reverse. Have we done that? Then write your master mix, and then write your nucleus-free water. I believe we have done that. So under the 20 microliter reaction now, for your template, we are still going to come back to that. So our master mix, whenever you buy your master mix, it's always written on it that the master mix is 5x. What that means is that you are going to divide the 5 by the total volume of your reaction. And since we are doing a 20 microliter reaction, that will be 20 divided by 5. That will be 4. So our master mix, we are going to take 4 microliters. So in front of your master mix, under your 20 microliter reaction, put 4. Write 4 microliters. That's 4 UL. For our primers now, whenever you want to do your PCR mix, although it varies, some people use 0.2, 0.3. It depends on the concentration they make initially with their primer. But since we did a 10 nanomolar, if you remember, 100 nanomolar, that we took 10 microliter and then 90 of our water when we were diluting our primer. So based on that, we are going to take 0.4 microliter of our primers. So in front of our forward primer, we write 0.4 microliter, and in front of our reverse primer, we write 0.4 microliters. I believe we have done that. Then for our template now, our template will now depend on your concentration and on your yield and on your purity. So let's assume we all had a great yield when we quantify yesterday. So our template, we can put 2 microliters there. But let's say you are now in a case where your DNA isn't that great, but it's still great. Then you can tweak your DNA template volume to 4 microliters. So the volume you use that for your template is dependent on the outcome you get when you quantified it. So I want to believe our DNA, our yield was great yesterday. So we are going to put 2 microliters in front of our template DNA. So you calculate that, you minus it, you subtract it from your 20. That's the amount of nucleus filled water we are going to use. We've calculated 4 from our, so we are going to do 2. That's 2 from our template plus 0.4 from our forward primer plus another 0.4 from our reverse primer, then plus 4 from our master mix. That's 6.8. So you subtract that from 20. That will be 13.2. So 13.2 is what you are going to write in front of your nucleus filled water. So we've gotten 13.2 for our nucleus filled water. So if you sum everything up, that's 20 microliters. So I said whenever you want to do this PCR, your water goes in first, followed by your master mix, followed by your primers. It doesn't matter which of the primers goes in first, whether your reverse primers or your forward primer. But because we are Nigerian, even me, when I know that it doesn't matter which one goes in first, but I always start with my forward. Then your template goes in last. The reason why we do that is when you start with your template and then your primers goes in, immediately your primer comes in contact with your DNA, it begins to read the temperature of that room as the temperature at which you want it to start annealing. So even before you put it in the thermocycler, the annealing will have started on the bench. The primer would have started its work. So when we now start with water, master mix, primers, and then our template like this, we put it inside the machine straight up. We put it inside the thermocycler. So that's why we start with water, your master mix, your primers, and then your DNA or your RNA. Then we set our thermocycling condition, our initial denaturation, then our denaturation, our annealing, our extension, our final extension, then old. What this old does is, let's say I put my machine, I have what's it called, 30 cycles to run. Then after my 30 cycle, when my 30 cycle, when the 30 cycle was already, done with it, I wasn't there. Maybe I had gone to do something else. So that temperature, 4 degrees Celsius, is going to hold it for me till I come back. And the already amplified DNA, we call them amplicons. So that's our workflow. Okay. So we've put everything in the thermocycler now. We can start our PCR. So most times it runs for an hour 48. It depends on the number of cycles. If you are doing qPCR cycle, qPCR and it's for 40 cycles, sometimes we use 2 hours on a single run, and sometimes an hour 47 minutes, an hour 48. It depends on the number of cycles. While we are waiting for our amplicons, we can talk about the types of PCR that we have. So the most common one is the conventional PCR. That's the standard PCR. It is the basic form of PCR. And for this one, the amplified DNA is detected at the end of the reaction, after all the cycles are complete. When you put now, what we are doing now, we are using conventional PCR. So we just put it inside and we believe that this thing that we have started though is actually going to amplify. It's now after it is done, we'll now make a gel. So the reverse transcription PCR, this one is used to amplify RNA and not DNA. So it makes use of an enzyme called the reverse transcriptase, and then the product you get from this one is called a complementary DNA, which is known as the cDNA. So you often use this one when you are working with RNA. But maybe you have an organism whose genetic material is RNA, but you are interested in DNA, so you have to get the RNA first, reverse it to cDNA, then from the cDNA, use it for your gene expression analysis. Then we have the PCR, that's the qPCR, that's the quantitative PCR. So this one measure the amount of DNA amplified in real time. And you can see the reaction as it progresses. You can see at what temperature does my sample one amplify, at what temperature does sample two. And this one, so the mechanism behind the quantitative PCR is that it uses a fluorescent detector. So whenever we make our mix for qPCR, we use a fluorescent dye or a fluorescent probe. Example of the fluorescent dye is the cyber grain. An example of the fluorescent probe is the Tachman probe. So this fluorescent will allow us to see the DNA as it's being amplified. And for this one, there is no need for post PCR process like running the product on gel electrophoresis. This is not needed because you are going to get your values in real time. Once it is done, you export your result into it comes in an Excel format. So it gives you your product for sample A, the CT values that you get, the at what temperature that, yeah, the sample expresses, and all the other information. So that is for qPCR. Mainly, that's the types of PCR that is very common. We have other PCR. We have nested PCR. We have digital PCR. We have multiplex PCR, but those ones are not common as these three: the conventional, the RT, and the quantitative PCR. So the summary is that PCR is an indispensable tool in genomics, and it enables rapid and specific amplification of DNA, which is fundamental to countless biological and medical applications.